All right, welcome to Chapter 9 on plants, and this is the video for Chapter 9.1, Transport in the Xylem. Okay, so we're going to start out with some comedy here, which is me trying to draw a cross-section of a leaf. So if we take a leaf and we kind of cut it in half, and I look at this tiny edge here, this is what I want it to look like. Let's see how close I can get. Okay, so on either side of the leaf, we're going to have a layer called the epidermis, okay? So I'm going to draw a layer on top, and I'm going to draw a layer on the bottom, but on the bottom, I'm going to leave a little gap. We'll talk more about that later. Okay, and of course, these are cells, so they would have all the things like nuclei and, Golgi and all that good stuff. Okay, so again, this is called the epidermis. Dermis meaning skin, epi meaning on, so literally on the skin. Okay, it's our kind of like protective layer here. On the upper side of the leaf is going to kind of be this layer, this waxy layer, and that's called the cuticle. We'll talk more about what that does later. Okay, on the upper side of the leaf, we're going to have a pretty tightly packed area of cells. Okay, and these cells are going to be the main photosynthesis workers. Okay, they are called the palisade mesophyll. Okay, and again, they're going to be on the upper side of the leaf, okay, so more apt to absorb those photons, that light, etc. Okay, now in here, we're going to have a region of sparsely placed cells, okay, and this region is what we call the spongy mesophyll. Okay, so again, meso meaning middle, so we're talking about the middle of the leaf, the palisade mesophyll and the spongy mesophyll. Okay, and this has a lot more air pockets in it. Okay, then the last thing that we're going to draw here, and that's not true, that was a lie. The next thing we're going to draw here is one of the veins, okay? Now, don't draw this next part because I'm not sure how well this is going to turn out. But the vein would actually like run through the leaf, so it's like a series of tubes, okay? But since I'm only looking at a cross section, okay, I'm only looking at it like head on. You'll see what real artists do in just a minute, okay? So this vein is actually full of a series of tubes, and their arrangement is going to depend on what kind of plant this is. But this is a vein. You've all seen them on leaves, okay? They run like this. There's different veining patterns, okay? And the vein is going to contain two types of tubes. They're going to contain the xylem, or xylem, and then the phloem. Okay, so two types of tubes in there. And now we have this awkward hole to talk about, okay? So the hole itself has one name, but on either side of the hole, we have these two specialized cells called guard cells. Okay, and then the actual hole is called a stoma, although you're more likely going to run into the plural, which is stomata. Okay, so in my head, this is what I was envisioning my drawing was going to look like here. Okay, we have our epidermis on the top and bottom. On the top, we have a cuticle. Here's my palisade mesophyll, tightly packed. Here's my spongy mesophyll, more airy. And here's that vein. We drew the head-on section, but it obviously goes through the leaf. Okay, so let's talk about these structures. So we have the cuticle, and the cuticle is what's going to protect the plant against water loss. So plants during periods of dry spells, can't just run down to the turkey hill and buy themselves a nice refreshing drink. Okay, they have to withstand varying weather patterns. So being sensitive to water loss is really important. And one of the ways that plants have adapted to do this is to, again, have this cuticle, this waxy layer that prevents too much water from leaving. Not all plants have a cuticle, okay? So if there's no cuticle here, then the epidermis is the top layer, okay? But that waxy nature prevents excess water from escaping. Okay, so now on to that epidermis, okay? Again, epi on skin, dermis, okay? So literally on the skin, on the periphery. And this is more of a protective, okay, layer, okay? So right here and right here. And it's made of flat, tightly packed cells on both sides of the leaf, okay? So there's not a lot of gaps in here, okay? They're very flat. And now on to the palisade mesophyll, which sounds like a really fancy neighborhood, but it's not, okay? So here's the palisade mesophyll. It's the cells near the top of the leaf, so that's super important, that are responsible for light absorption and photosynthesis. And that's why they're on the top and not the bottom, so that when light comes in, okay, they're kind of, other than the epidermis, the first cells that things run through. So let's see, this is going to contain a whole lot of chloroplast because that's what's going to be needed for this photosynthesis that these guys are really taking care of. Okay, so the other meso or middle part is this spongy mesophyll layer, and it's exactly what it sounds like. There's a bunch of air pockets in it. Okay, so there's sparsely placed cells, and there's channels here for gas exchange. And so that's why it's full of air pockets. Those air pockets are for gas exchange. Okay, so we've got we've got two gases, okay, that are really coming in and out of these cells, and we need a space for that kind of exchange. Okay, now the stomata and the guard cells kind of go hand in hand, and we're going to talk a lot about them later. So here they are, okay, the guard cells are on either side, the stoma or stomata plural, okay, are the holes in the middle. And this is the place where carbon dioxide is going to come in. Notice that they're on the bottom of the leaf, okay? That's because it's cooler down there, okay? So this is also a site of water loss. So if we want to control how much water is lost and prevent excess water loss, we don't want those holes to be up here where it's super hot and we're going to be getting a lot of absorption of radiation that's going to heat things up. We don't want that, okay? We want those holes down there on the bottom so that if we have to open them to let carbon dioxide in, we hopefully don't lose too much water in the process. Okay, now I should have prefaced this by saying that here we are talking about what we call vascular plants. Okay, not all plants are vascular. Vascular means that you have vessels that carry things. So in humans, our vascular system includes arteries and veins. Okay, in leaves and in plants, we say that they have veins, okay, but really those veins contain two types of tubes or two vessels, okay? It contains the xylem and the phloem. We're going to talk a lot more on that later. We do need to know that they're going to occur in the middle of every leaf, okay, so that they can service all leaf cells. These guys are going to carry in the water that we need, and they're going to carry nutrients around, okay? So they're just like the vascular system in humans. Okay, so how does water move throughout a plant? Okay, now plants can't drink things through a straw like you can, okay? What you're actually doing involves like muscle contractions, okay, and physically pulling water up there. Plants can't do that. They don't have muscles, okay? So instead they use a process called transpiration, okay? Which again is nothing like drinking through a straw, okay? This is where water travels from the root of a plant to the leaves, okay, and it exits through the stomata, those holes on the underside of the leaves, as water vapor and gas. So we know that some of the water that plants take in okay, is consumed through this process of photosynthesis, but 90% of it is actually lost via this process called transpiration. Okay, so this is a super important process, okay, for plants. So it kind of sounds bad that we're losing the water. It's not all bad. Okay, the first thing it's going to do is it's going to carry water to various parts of the plant. Okay, we know that water is only absorbed by the roots. It doesn't matter how much it rains on a plant. Okay, the leaves have no way of absorbing that water. Okay, all of the water comes into the plant via the roots. Okay, so the process of transpiration carries water to various parts of the plant. Okay, along with that, it's also carrying minerals that are dissolved in the water, and it can actually function as a cooling mechanism for plants, just like sweating does in people. Okay, so we may see this process as starting from the bottom up, but realistically, it's starting from the top down. Okay, so this water that's being lost through evaporation is replaced by stuff in the plant, which is replaced by the roots absorbing more water. Okay, so again, the veins of the plants are involved in the transport of water or transpiration, but only a certain section of the vein. Okay, so within the vein, I'm going to find two different types of structures, the xylem and the phloem. The xylem carries water and anything dissolved in it up through the root of the plant. It's a one-way train. The phloem, on the other hand, okay, is going to carry things like sugars and other metabolic products from wherever they're produced to where they're stored. So that could mean from bottom to top. It could mean from top to bottom. It could mean side to side. So that direction is dependent. Okay, the xylem, on the other hand, always carries things upwards. Or I should say away from the root. Obviously, I know some of you are going to come back to class and say, what about leaves that are side to side? Okay, anytime we're going from root to leaf, okay, it doesn't matter, okay? That's eventually going to involve some kind of upward movement. Okay, so let's take a look at how to draw the xylem. The xylem is going to be made of two different cells, and the first type of cell we're going to be drawing is called a tracheid. Tracheids are kind of long and slender, and they have these angled tips to them. And the reason why is because this next one that's like right on top of it has a very like complementary shape. Now, this is a dead cell. There's no nucleus or like any living parts, okay? And it's hollow. In between them, it's got these little like pits. Okay, so I can see this is going to be fun to draw. Okay, it's got these little pits or holes, okay, that are going to allow things to pass from one to the other. Also, in the side of it, it's going to have pits, okay, to help things move sideways. Okay, so these are long and slender. The other kind of cell we're going to find in the xylem is what we called a vessel element. Okay, and vessel elements are a little larger in diameter. Okay, and they're going to kind of butt up to one another like so. And again, the ends of them are going to have pores so that things like, I don't know, water can pass from one cell to the next. Okay, so the ends of them are going to have these pores in them. Now, the other interesting thing about the vessel elements is they have this stuff that goes around them, kind of in a spiral, okay? I don't know how I'm going to draw that. Let's just pretend this looks like some kind of like a candy cane stripe, okay? And this stuff here is called lignin. We'll talk more about that later, okay? In the meantime, I forgot to label these pores. Okay, make sure you do that. Okay, so that we can make sure that we remember that stuff can travel up and down from cell to cell through those pores, but it can also travel side to side out of these pits here in the trachea to the cells that are right next to it. Okay, so here's what my drawing was supposed to look like. Here is so long skinny tracheids with those angled tips, okay, and then the vessel elements a little bit wider, okay. This shows it at an angle. They don't always meet at an angle like that. This also doesn't have the lignin on it, but we'll see more of that later. Okay, so they are both made of dead cells. That is super important. The xylem contains dead cells. That's going to be different for the phloem, which we'll get to in the next chapter. So tracheids have tapered ends. They fit together. Water moves between them. The vessel elements have thick walls with pores in them, or pits, okay, that allow water movement in one continuous column. So we can kind of see in this micrograph here how much larger in diameter the vessel elements are than tracheids. Okay, so quick review here on plant cells. We know their cell walls are made of cellulose, okay? It's a polysaccharide made of glucose monomers. It's linear. Well, this is all, should be all review. The vessel elements have an additional cell wall, okay, that's made of another complex carbohydrate called lignin, okay? And so here's kind of that spiral, okay, lignin that I was trying oh so hard to draw on my drawing, okay? And this stuff is super strong, okay? So it helps to strengthen the wall, okay? It also helps to make it a little bit more waterproof so we can get the water all the way up to those really tall plants to their most upper regions, okay? And it's a very good system for support. So you got to remember that plants don't have skeletons, so they rely on things like their cell wall, this lignin, and Turgor pressure, which we'll get to later, to help them stand up straight. Okay, so now on to one of the more fascinating structures of the leaf, which are the stomata. And again, those are the openings in the leaves, and they are surrounded by two guard cells. 1, 2, that number is important. Okay, so they let water out, which can be a good thing, like if the plant needs to cool off, but in general can be a bad thing. Excess water loss is never a good thing. But they're also essential because they let carbon dioxide in, okay? So in order for plants to get the carbon dioxide that they need for photosynthesis, these things have to be open, okay? So they can open and close, but any closure has to be temporary, okay? Or we're not going to be able to get that carbon dioxide that we need to begin the Calvin cycle. Okay, now this whole system works on something called Turgor pressure. Okay, and Turgor pressure is the pressure that's exerted on the inside of the cell due to water. Okay, so water is going to fill up this central vacuole of this guard cell. Okay, and that's going to determine whether the guard cells are open or whether they're closed. Okay, and so here's how that works. Okay, when the plant has a lot of water availability, okay, water is going to enter the central vacuoles. It's going to cause those central vacuoles to blow up and to exert a lot of pressure, Turgor pressure. I shouldn't have used that color for that, I'm sorry. Okay, it's going to exert a lot of pressure on the plant cell itself, okay? That Turgor pressure is going to cause the plant cells to become curved, okay? So these guard cells are going to curve due to all the pressure on the inside. When they're curved like that, it opens the stomata. When a plant cell does not have enough water, the plant doesn't have enough water, okay, then water is going to exit those central vacuoles, okay? It's going to cause a decrease in Turgor pressure, and these guard cells kind of go limp, okay? There's no more pressure in here. when they go limp, they kind of close that opening, okay? And when that opening gets closed, then nothing is allowed to get in anymore. Okay, so that kind of makes sense. If this opening is open, if the stomata is open, water can escape, but it's not a big deal if you have a bunch anyways. If we're low on water and water leaves that central vacuole, it in turn closes the stomata, helping the plant to conserve water. Okay, so we know this has something to do with water, but why is that water moving in there? Okay, so in the presence of light, there's an ATP-powered membrane pump, okay, that's present in the membranes of these guard cells that is going to actively pump potassium ions into the cell, okay? That's going to cause a difference in concentration, okay, and those cells have no way of diffusing back out on their own. Okay, so nearby water is going to move into the cell via osmosis due to the change in potassium concentration. Okay, that's going to cause, again, this central vacuole to fill up. Okay, then this Turgor pressure gets exerted on the cell, making it this jelly bean type shape and opening up the stomata. In times of a drought, okay, plants are going to produce a hormone that causes potassium ions to leave the cell. So this is a hormone action here. Okay, and when that happens, okay, to maintain the osmotic balance, water is going to leave also. Okay, now if I can only convince my pen of that. Awesome. Okay, when the water leaves, okay, the Turgor pressure inside of the cell is going to decrease. The cell is going to become flaccid or limp, okay, and it's going to close that stomata. Okay, so transpiration and the stomata and the guard cells, they're all linked in what's called the cohesion-tension theory of plant fluid movement. It really starts from the top of the plant down. It's easy to think of water movement as going from the roots up, but that's not how it happens. Okay, so the first thing that's going to happen is water is going to exit through the stomata, okay, and it's going to evaporate. And this is due to the fact that there's more water in here, okay? These awful blue marks I'm making must in my brain represent water. So there's a higher concentration of water in here than out here. It's really wet, it's really dry. Water is going to move from where there's a lot of it to where there's not as much. So some of that water is going to evaporate. Okay, so the water that's lost from the evaporation in the stomata is going to be replaced by water that's in the vessels of the xylem. Okay, so here's the xylem. Okay, that water that's in the xylem is going to replace the water that we just lost through the stomata. Now, we talked about this a million times. Oh, why are cohesion and adhesion important? Well, here's finally where we get to learn this, okay? The cohesion between water molecules kind of pulls that entire column up. So follow me here for just a second. This water molecule leaves, but it's really cohesive with this one, who are really cohesive with these, who are really cohesive with these. And so we can actually trace this line of attraction or cohesion all the way up. And you can imagine how if this water molecule exit, it pulls the entire column of water up through the xylem. Okay, and this column of water that's in the xylem, so this here, okay, is maintained by cohesion and adhesion. So again, what we just talked about on the last slide is cohesion is going to keep water molecules stuck together. Like this one's saying, hey, wherever I go, you follow me, okay? Like dragging your little brother through the mall. Okay, adhesion is pulling that water against the force of gravity because of the attraction between the sides of the xylem and that water molecule. Okay, so kind of pulling it up and then keeping it together. Okay, so all this pulling and tugging is going to cause the force known as tension, okay? So tension, if you can imagine like a tug of war and like pulling on a rope, we can feel that tension in the rope. Okay, the same thing is going to be happening in the water column inside the xylem. There's going to be a lot of tension, okay, as we're literally pulling water molecules up through the xylem with the forces of cohesion and adhesion. Okay, so we know how water exits through the leaf, evaporation from the stomata. We know how it travels up the xylem, okay, as a water column. But remember, it all comes from the roots, okay? So in the root cells, we have water. It's called in the root cortex, okay? And that tension that we've established in our water column is going to pull the water out of the roots, okay, and into the xylem. So it all sticks together. And then finally, okay, that tension, okay, that pulling motion is going to suck water from the soil into the roots, okay? So again, it starts from the top downwards, okay? And I know that's kind of hard because the water itself is actually moving up, but the only reason it's moving up is because there's some action happening, okay, from the leaves, which is pulling water through the xylem, which pulls water from the roots, which pulls water from the soil into the roots. Okay, so at this point, you should be able to draw a diagram of transpiration. Okay, so at a minimum, you should be drawing, obviously, leaves, okay? Stomata, well, that's embarrassing. Okay, I should see a xylem in there. Okay, we should have roots, we should have soil, okay, we should have water, don't want to forget that, okay, and also there should be all kinds of arrows happening. Maybe you even label forces like cohesion, adhesion, or if you want to impress someone like, I don't know, your teacher, maybe tension, okay, so make sure you draw your own diagram. Okay, so let's talk about the roots a little bit. Okay, so we know that roots are in general like underground. Okay, and their job is to absorb minerals and water from the soil. So they have a couple really cool adaptations for that. One of which is that they usually have many branches. Okay, and then they also, within each one of these roots, okay, have these tiny little things called root hairs. Okay, and just like the microvilli on your intestinal cells, these hairs are here to increase surface area for absorption. So you know how we always say, like, if you're asked about a process, you don't know what the answer is is usually ATP or enzymes. And if it's about the structure, it's usually to increase surface area for absorption. Okay, here's another case where that's definitely true. Okay, so that water that's in the soil, well, hopefully, unless you're in some kind of a bone-crushing drought, is going to move into the root for two reasons. Okay, first of all, we talked about the tension from the water column. So as water is evaporating here, it's pulling up water from the xylem that's pulling water out of the roots, and that tension pulls water from the soil into the roots. Okay, so the tension, there's one reason. The other reason is sheer like osmotic balance, okay? So within root cells, okay, they will often, wow, what just happened there? Okay, they will often have a much higher solute concentration on the inside of their cell than the soil around it, so that water will enter those root cells. Okay, so my students, you may have some fond memories of looking at root cells and calculating the mitotic index by identifying mitotic cells and comparing them to the numbers of non-mitotic cells. And we were using that to determine which part of the root is actually growing, okay? So I told you that that activity would pay off once we got to plants, and here we are. Okay, so let's draw a root. Okay, so roots are three-dimensional, tubey things, okay, and they kind of come to a point like this, right? So kind of like a pointy-ended test tube. Okay, and the very outer layer, oh, that's a bad picture. Oh, well. Do you understand what I'm getting at here? The very outer layer, okay, is the epidermis. Okay, so there again, we're talking about that outer layer of cells. In the middle, going all the way through the inside of the root, oh my gosh, is something called the vascular cylinder. Okay, so the vascular cylinder is exactly what it sounds like. It's a cylinder. It actually runs all the way through the plant, okay, or the root rather, but we're looking at the outside of it. So you're going to have to imagine as if this were going all the way through. If my drawing is so terrible that you just can't imagine that, you're welcome to draw it going all the way through. Okay, it's exactly what it sounds like. It's a cylinder made up of vascular tissue, so we're talking xylem and phloem. On the outside, up here towards the top of the root, okay, we're going to find root hairs. And down here at the bottom, okay, we're going to have two structures. At the very bottom is what we call the root. Oh my gosh, this pen though. I hope you're having some fun with this. This is the root cap. Okay, we'll talk about what that does later. Okay, and then a section here called the apical meristem. Talk about that later too. Okay, now all the rest of the actual root, other than the cap and the apical meristem, can be divided up into three sections. So we have a section at the top, a section in the middle, and a section at the bottom. This first one is called the zone of maturation, followed by the zone of elongation, and the zone of cell division. And I love these because they're exactly what they sound like. Okay, we'll talk more about those zones in just a little bit. Okay, so let's talk about what's going on here in the root. Okay, root hairs are going to help us with absorption. Okay, so absorbing minerals or water. Okay, increasing the surface area. The root cap is there for protection. So we know that roots grow downward. Here's my tree, here's the roots, okay, here's the ground. Look how amazing that is. Roots are growing downwards, and they've got to push through a whole lot of things like rocks and dirt and other stuff. So that root cap is going to protect it. And then we have those three root zones. Okay, so the zone of cell division, okay, is where we're going to find a lot of cells that are actively undergoing mitosis. Okay, we like it when they sound like what's actually happening. They're called meristematic cells, okay, because they're not specialized and they're dividing rapidly. So just like stem cells in a human embryo that are divided rapidly, don't have a specialist or a specialization yet, okay, these meristematic cells, okay, are undifferentiated. They're dividing really rapidly. If I look at them under a microscope, wait a minute, didn't you do that? I think you did. You should find a lot of cells in mitosis and a high mitotic index. Okay, so maybe you should add that to your notes. Okay, a high mitotic index. Okay, a lot of those are undergoing cell division. So this is our area of actual growth. Okay, this is where the growth is actually going to happen, new cells. Okay, then we have the zone of elongation. Okay, this is where the cells are actually just growing in size. So when we talk about our cell cycle, we have interphase, and then we have mitosis. Within interphase, okay, we have G1, okay, and this is cell growth, so the zone of elongation. S, okay, that's DNA replication. G2, preparation for cell division. And then we have mitosis and cytokinesis. So this is our zone of cell division, okay? And then this G1, okay, cell growth is happening in the zone of elongation. So the cells are actually getting longer, they're getting bigger. Okay, and then finally, okay, all the way at this end of the root up here, okay, we have what's called the zone of maturation. And this is where those cells really become specialized. Oh my gosh. Okay, specialized and differentiated and part of the plant. Okay, so they're maturing into cells that are actually going to be functional. And here again is that vascular cylinder we were talking about. Okay, because once these root hairs help absorb water from the soil, okay, we need that to actually travel upwards to the rest of the plant, okay, into the xylem. Okay, so something like that. Okay, so when we were labeling the root, we talked about the apical meristem. So we know the meristem is kind of like that bunch of stem cells that just divides and divides and divides to make new cells. Okay, apical meristems are going to be down here at the bottom of the root, and they're going to help the root elongate. Okay, so help grow in length in a vertical direction. A lateral meristem is going to help it increase its diameter. It's going to get fatter. Okay, so that corresponds to primary growth, which is vertical growth. Okay, and lateral growth, which is getting bigger in diameter. So some of you are still growing taller. Me, on the other hand, I'm doing this, okay, just like plants do. Okay, their roots have both an apical and a lateral meristem. Okay, so we know that pure water doesn't exist in nature, right? That it's such a good solvent that anything that it's around, it's likely to dissolve some kind of minerals. So in addition to the water absorbed by the roots, okay, the roots are also going to pick up several minerals, like inorganic substances from the soil. That's what all these funny letters are. This is going to happen in one of three ways. Okay, so the first one is going to be diffusion. Okay, so this is the simplest scenario. Let's say that in the soil there's a really high concentration of a certain mineral. I don't know, who cares? Make one up. Magnesium, great. Okay, and in the root there's a really low concentration of that particular mineral. Okay, then the mineral is going to diffuse into the roots from an area of high concentration to low concentration. So that's a passive process, no need for any kind of energy input. Okay, the next is going to be through those really cool symbiosis that many plant roots have with fungi. Okay, so here's just a naked plant root. Okay, I see some root hairs, but it's only able to absorb things, okay, at its surface. Okay, so like all along here. Let's say that it's able to accomplish this symbiotic relationship with fungi, and fungi have these hyphae which are able to absorb things. Now look at all of the surface area that this root now has available for absorption. Okay, so this increases the surface area in times when an increase in surface area is either needed or it's advantageous, like low nutrient soils, etc. Okay, and the last one we're going to talk about is active transport. Okay, so here's my root cell, and my root cell already has a pretty high concentration of a certain mineral, okay? But I want more. I'm a greedy cell. So out here in the soil, this low concentration of mineral ions exists. In order to get these guys to come join the party in here, I'm going to need to add energy in the form of ATP to power this protein pump, okay, because I'm going against the concentration gradient, okay, from an area of low to an area of high concentration. Okay, so the last part of this chapter is all about how plants deal with some adverse conditions, okay? There are some things that can alter or affect how much water we're losing from these plants. And of course, the amount of water that we lose is related to the rates of transpiration and the rate of movement through the xylem and root absorption. Everything is all related, okay? And so the first thing here is light intensity. Okay, so the more the sun is shining, okay, or the higher the light intensity, okay, the more heat I'm going to generate, okay? So when this solar energy is absorbed by the plant, it gets radiated as heat energy. I don't know what all these arrows are supposed to mean, but they're there. Hopefully you're getting something out of this. Okay, so that light intensity causes the leaves to become warm, and that means transpiration is going to speed up. Okay, so end of story, more light equals more transpiration. Okay, so the next one's a little bit more difficult to understand. So if you remember when we were looking at this diagram, water is moving out of the stomata because there's a lot more water in the plant than there is in the atmosphere. Okay? So if I were to increase the humidity around the plant, okay, then transpiration is going to decrease because now there is no need for water to move out here, okay? Water's only moving when there's a difference in water concentration. So if I increase the amount of water vapor in the atmosphere, okay, this water won't evaporate so much. Now, if I add wind in there, then things are going to get a little interesting. So I just said we had all this water out here, which meant that this water in here didn't need to go anywhere. Well, if some wind comes along and knocks all this water away, okay, now I have a difference in water concentration, and this water is going to leave. It's going to transpire. So wind generally causes transpiration rates to increase. Okay, so we already talked about light intensity. Okay, now let's maybe back up a step and talk about just temperature. So just with humans, if I increase the temperature, okay, and we need to maintain our constant body temperature of 37 degrees Celsius, then what's going to happen is that our bodies are going to send water to the surface of the skin. Okay, that water is going to evaporate, okay, and it's going to carry some of our body heat with it. So it's a method of cooling down. Transpiration is the same, okay? When this water leaves, it does so as water vapor, okay? And so that evaporation process takes some of the heat from the plant with it. So if I increase the temperature, that's going to increase how often or how much water is transpiring, much like if I increase the temperature, it's going to increase how sweaty someone is. Okay, so where else am I going to find water? Not just in the atmosphere, but in the soil, okay? So the more soil water I have, okay, or the more moist my soil is, okay, the more often transpiration is going to happen, okay? So remember, when we don't have a lot of water coming in, those guard cells kind of become flaccid and closed. When we have plenty of water, it causes the guard cells to kind of open up and that stomata opens, okay? So if I have a lot of water from the soil, okay, entering from the roots and then all the way up into the leaves, that's going to cause the stomata to open, which is going to cause transpiration to increase. Okay, and the last thing that seems to affect transpiration rates is carbon dioxide concentration. Okay, so if I increase the carbon dioxide concentration, okay, these stomata tend to close. Okay, and if these stomatas close, then no water can get out, so transpiration is going to decrease. That may have something to do with, I'm thinking about our photosynthetic rate graphs. I'm being attacked by a small dog right now. I just want you to know how difficult this is to make this. And carbon dioxide concentrations. And we know that it levels off at some point because of all the enzymes involved in the Calvin cycle are already busy. But I wonder if those enzymes can somehow sense the fact that we don't need any more carbon dioxide here, and so they close the stomata, and if the stomata is closed, no water can get out. I don't know. Okay, I just know that they close. Okay, so if we know that there are certain environmental conditions that can increase the rate of transpiration, and plants don't want to lose too much water, okay, then plants that live in those gnarly conditions are likely to have some really cool adaptations so that they can still survive. Okay, thank God this video is almost over because I have someone here who really doesn't want me to spend time doing this anymore. Okay, so let's talk about these plants that really have to work for their survival. We call them xerophytes, okay? Xero meaning like an arid place. Okay, those are plants that are well adapted to survive in like dry climates or arid climates. Okay, those include our cacti and our succulent plants. And then we have halophytes, okay, halo meaning salt, and those are plants that are well adapted to survive in high salt levels, okay, or with water that has a high salinity. Okay, so how do xerophytes, plants that live in like, let's say a desert, okay, how do they prevent water loss considering how dry and hot and windy those deserts are and how little water they receive? Okay, well, the first thing that they do is they have small, thick leaves, okay? So the fact that they're small and the fact that they're thick means that they have a lot less surface area, and that's good because the surface is covered with stomata on the bottom side, okay? And it's also an area for it to get like very hot and dry air contact. So if we can limit surface area, that's good. They also, just in general, have fewer stomata, okay, so that water can't evaporate as much. They also have their stomata in pits. This part is so cool. So instead of having the stomata down here that's in contact with all of this dry air, they have a pit on the underside of their leaf, and they have the stomata in here. And that's so cool because water can actually like accumulate in here. And by water, I mean humidity. So this air here in this pit is a lot more humid out here, which kind of tricks the stomata into thinking that it shouldn't let through any water, okay? Because then this water concentration and this water concentration are a lot more similar than this water concentration and the air out here, okay? So these sunken stomata are pretty cool. Okay, so in those stomata, okay, those sunken stomata, they have these little hairs, okay, and that traps the humidity that we were just talking about so that there's not a huge difference between the water out here, okay, and the water level in here. So it limits how much water is leaving by tricking those guard cells into thinking the air out here is actually really humid. Okay, they're also covered in a thick cuticle, okay, a thick waxy cuticle to prevent water loss. And sometimes they just give up, okay? They become dormant in those drier months so that they don't need as much water, okay? So remember, water is one of the necessary reactants for photosynthesis, okay? And so if we just go dormant, then we don't need as much water. Okay, and one of the other things that these cactus or these desert plants or xerophytes are really known for are their shape, right? So they're kind of like hollow. Here's an aloe plant, same thing. They have this mechanism for storing water in their big, fat, fleshy stems. And so on the odd occasions when they do receive rain, they can kind of store that water in here like a little like savings bank, okay, for those dry months. Okay, so halophytes, okay, those plants that live in like salty water areas, okay, also have some adaptations to help keep them alive in their environments as well. So they also store water in their fleshy stems, but they do it for a different reason. It's not because they're worried that it's going to be dry later on. It's because they need to dilute that salt, okay, that's in the water that they're bringing in through their roots. A lot of them also have special salt secreting glands. So they absorb salty water, but then they get rid of some of the excess salt, okay, through special cells in the undersides of their leaves. Okay, they're also good at taking the excess salt and like putting it into special vesicles in the cells to keep it away from the rest of the cells. Okay, so kind of like a prison for salt inside the cells. We don't want a ton of salt in the cytoplasm because it messes with the osmotic balance. So if we can compartmentalize it in those vesicles and keep it away, then that's a good thing. And a lot of them do the same thing that the xerophytes do, just reducing their surface area, so these shorter, thicker leaves and other parts to decrease the salt exposure. They don't need a ton of surface area for absorption of water. They live in salt water. Okay, so in that case, a lot of surface area is bad, so they can actually decrease it so they don't absorb as much salt water to begin with. Okay, so this one I'm going to show you this graphic and then I'm going to let you think about this and answer this on your own, okay? Which is, what is the relationship between atmospheric carbon dioxide and stomatal density? So this is one of the new burgeoning fields in climate change research. So researchers have actually been collecting plant samples and they're noticing that stomata density has been changing over the course of the last century or so. Whereas before plants had a lot of stomata, okay, now they have less stomata, and it's linked to the amount of carbon dioxide in the atmosphere. So it's kind of another one of those indicators that we can use to show that global atmospheric conditions are changing, okay, that carbon dioxide levels in the atmosphere are changing and it is affecting some plant physiology, or at least we can use the plant physiology to prove that these numbers are in fact changing. Okay. So one of the things that we know about food is that it all comes from plants, okay? So even if you eat a chicken for dinner, probably not a whole one, but a part of one, that chicken ate some kind of plants. It all goes back to primary producers, okay? Now, we know that transpiration rates change with the temperature. So here's what's been happening to the temperature over the last few years. Okay, I want you to think about how that increase in temperature increases transpiration and what on earth that could possibly mean for global food production. Okay, and you should have an answer for that on your own when I see you. And that'll do it for chapter 9.1 on transport in the xylem.